Well, first off, I want to thank the organizers for letting me come here today and share with you how we're using this next generational nanomedicines to develop vaccines against infectious diseases. And particularly, I'm going to talk about a couple stories today. The first is vaccine development against the Zika virus, and there's some stories that were published, and also some new data coming out of our lab on another flavivirus, dengue virus. And so, to start with, I'm going to give a brief background of the Flavivirus family. And so this is a family of well-known viruses. For example, we've all heard of West Nile virus, yellow fever virus, and also dengue virus and Zika virus, which are the topics of today's conversation. So these viruses are transmitted through the bite of a blood-feeding insect, mosquitoes or ticks. And up here on the top, these viruses can be transmitted through a number of mosquito species. And these mosquito species determines the geographical range of these viruses. For example, West Nile virus is carried by the Culex species of mosquitoes. And this mosquito species can be found at higher latitudes. And this is why we have West Nile virus up where I live in Chicago. However, and counter to that, these viruses in the yellow fever clade shown here below in the orange, these viruses are transmitted by the Aedes species of mosquitoes. And these mosquitoes are found more in the tropical and subtropical regions of the world, which is where we see these diseases from these viruses are endemic. So this family of viruses, these are envelope viruses with a positive sense single-stranded RNA genome of 10 to 13 kilobases. And this genome encodes for a single open reading frame, which is proteolytically cleaved to the individual proteins. And you can see here on top is the viral genome, and there's 7 non-structural proteins and 3 structural proteins, the capsid C, the membrane or premembrane, prM, and the envelope protein. And down here on the right is what this virus looks like, or an artist's rendition of a virus. You can see the genomic RNA there inside in the middle of the virus. And this genomic RNA is surrounded by the capsid protein. On the outside, we have a lipid bilayer, and we have the membrane and the envelope proteins are embedded within this lipid bilayer. And on the surface of the protein, what we have is the E protein or the envelope protein dimers, which coating the entire surface. And for the purpose of vaccine, we want to generate antibodies, which are going to bind strongly to this envelope dimer and inhibit the virus from infecting neighboring cells and neutralizing the virus. So again, the first part of this talk is going to be about Zika virus, and next we're going to go into some work on dengue virus. So first, Zika virus was first discovered in 1947 in the Uganda Virus Research Center in the Zika forest. And there was a couple human cases associated here in Africa in this time frame. Then in 1970s, in the late 70s, the virus was discovered in Southeast Asia, and there was a few human cases reported here too. But prior to 2007, this was the extent of Zika virus in the world. There was 14 documented human cases. Then the virus began to spread across the Pacific. So in 2007, there was an outbreak of Zika in the Yap Islands of Micronesia. And then in 2013, the virus reached the French Polynesian Islands. And by 2015, the virus reached the shore of Brazil. And from here, the virus spread throughout South America, Central America, and even up into North America. And from 2015 to 2017, there was more than 500,000 human cases either documented or suspected. And the virus is predicted to remain endemic in the Americas. And that's due because the vector is the Aedes aegypti mosquito. And this is the range shown here in red of where the mosquito vector persists. And it's predicted that the virus is going to co-circulate with dengue virus, which is also spread by the Aedes aegypti mosquito. And we'll see periodic outbreaks of Zika virus in naive populations for the foreseeable future. So most people infected with Zika virus show little or very minor symptoms. So some of these minor symptoms include the classic febrile illness. We see fever, rash at the site of infection, joint pain, conjunctivitis. But a subset of individuals will develop more severe symptoms. Some of these more severe symptoms can include neurological disorders such as Guillain-Barré syndrome. But by far, the greatest threat to human health is to the developing fetus in the pregnant individual. So a pregnant individual infected with Zika virus, the virus can be spread to the placenta, where you can have damage to the placenta and placental insufficiency. The virus can also be spread into the developing fetus, and the virus can infect the neurological development within that developing fetus, leading to intrauterine growth restriction, ocular abnormalities, and fetal demise. And it's important to know that we don't know what the long-term outcome of Zika virus damage to these fetuses. This is going to take years of research, and there's cohorts which are enrolled right now which are continually being tracked. So the actual damage of the Zika virus to cognitive abilities in the long term is not known. So in 2015, there was a virus pandemic with novel disease signatures, and this led the WHO Health Organization to declare a public health emergency in February 2016. And in this public health declaration, they called for the rapid development of antiviral vaccines and therapeutics. Well, we have a rich history, we the scientific community have a rich history of flavivirus research and vaccine development. We have vaccines for Japanese encephalitis virus, for yellow fever virus, and a lot of research done in the dengue virus field and West Nile virus field, amongst others. So we have established correlates of protection for related viruses, and we know what some of the strongest neutralizing epitopes are for these viruses, or related viruses. And this led to the rapid induction of several different vaccines. And there's more than 45 different vaccines which are under development, or various stages of development for Zika virus. And several of these have either completed early phase clinical trials or are in early phase clinical trials right now. So some of these platforms include the purified inactivated virus, the live-attenuated virus, the subunit vaccines. This is just recombinant proteins, either the envelope protein or specific domains of the envelope protein, such as domain 3. But there's also several vaccine platforms which are including the viral antigen encoding. This includes the DNA, mRNA, and adenovirus vaccines, which encode for the membrane and the envelope genes of the virus. There's also a measles vectored vaccine, which encodes for the non-structural protein 1 of the virus. But of course, today we're going to talk about the mRNA vaccines and the development of a novel mRNA vaccine using the mRNA lipid nanoparticle gene therapy platform. And so with this platform, we simply have the mRNA which encodes for our viral genes, which is capsulated in a lipid nanoparticle. And we're going to hear some more about some of these nanoparticle structures later on in the second talk today. So there are several advantages that this mRNA platform presents over other gene therapy platforms. One is the rapid, cost-effective mass production. It's very easy to generate large quantities of RNA in vitro in a test tube. The second is this platform is non-infectious. This makes it safe for any immunocompromised patients that may have any adverse events from traditional vaccines or live-attenuated vaccines. Another advantage is this is not an integrating platform. The mRNA can't integrate into the host genome into an oncogenic site and lead to any unintended consequences. And also there's no immunological memory against the mRNA, so we can have repeated vaccination with the same platform into an individual. So we made a mRNA-LNP Zika vaccine, and this vaccine is simply the membrane and the envelope protein are expressed behind a signal peptide. And this open reading frame is flanked by a 5' and 3' untranslated regions. And so this RNA, it can be in vitro synthesized, then we enzymatically add a 5' cap1 structure and a poly A tail at the 3' end. So this RNA looks like the host mRNA. With a couple of key modifications, we're using modified nucleosides. We use pseudouridine in place of uridine in the 5-methylcytidine, and this has been shown to limit the innate immune stimulation, which is important for transgene expressions. So we get a robust expression of our genes of interest shown here. So we generate this mRNA, and then this is encapsulated into a lipid nanoparticle, and this is injected intramuscularly. And so the encoded mRNA is then translated at the ribosome within the ER. The envelope protein and the M protein are folded into this virus-like particle, which is shown here in the top right. And this virus-like particle essentially consists of a lipid bilayer with the membrane protein, the M protein, embedded in that lipid bilayer, and on the outside is the E protein surrounding the entire thing. So it's essentially, it looks like a shell of the envelope protein, and the dimer of the envelope protein. And so this viral-like particle is secreted from the cell, and the host mounts an immune response against this viral-like particle. So the viral-like particle looks very similar to the virus. It has the same tertiary and quaternary structures of the virus. So the humoral response initiated against this VLP is similar to the humoral response initiated to an actual viral infection. So we should generate similar neutralizing antibodies as to an actual live virus infection. Now, to study this vaccine, we're using the mouse model of Zika virus pathogenesis. So Zika virus naturally infects primates. And so to get the mouse model to work, we must make the mice immunodeficient. And we have several ways we can do this. We can use genetically knockout mice, which includes the AG129 mice, which lack both type 1 and type 2 interferon signaling. We use type 1 interferon receptor deficient mice, the IFNAR1 knockout mice. We can also use a monoclonal antibody which inhibits the IFNAR signaling, so interferon signaling. So in these immunodeficient mice, we infect the mice with the virus, then the virus spreads systemically, we get viremia, and the virus seeds some of these immunoprivileged sites, including the central nervous system and the male and female reproductive tract. The virus can replicate to high titers in these susceptible tissues, and this can lead to death of the animal. So the first thing we did is to just test our vaccine efficacy in a immunocompromised AG129 mouse. And so we tried a high dose and low dose of the vaccine, either 10 micrograms or 2 micrograms, either a single administration or administration and then a boost 3 weeks later. And at 42 days post-vaccination or post-initial vaccination, we analyzed the serum and then infected the mice with the virus. So here on the bottom left, we're looking at serum neutralization. This is at 42 days post-vaccination. And you can see what this graph is showing here on the left is at high levels of the virus, high levels of serum, so undiluted serum, we have complete neutralization of the virus. But then as we begin to lower our serum dilution, we see that we're no longer neutralizing the virus. And so what we're able to calculate here is the EC50 value, or the value at which 50% of the virus is neutralized. And this is quantified here in the middle graph, looking at serum neutralization. You can see that mice that had received 2 doses of either the high dose or the low dose of the vaccine both exhibited robust antiviral antibody titers. And mice that had received a single dose of the high or low dose of the vaccine induced antibody titers, but not as robust as the 2-dose regimen. And when we challenged these mice, looking at survival of these mice, you can see that the placebo mice here in black that did not receive the vaccine, these mice also come to viral infection, but mice that were vaccinated were protected. And this protection correlated with the levels of neutralizing antibody titers. So the vaccine's inducing high antibody titers, and it's protecting our mice. So we also looked in the susceptible tissue. So here we're giving a high dose or low dose of the vaccine in a prime boost strategy. And again, we see high levels of viral neutralization with both these strategies shown in the top right. And then if we look in the susceptible tissue in the CNS in the brain, central nervous system in the brain, or within the female reproductive tract, we're reducing viral titers by log levels. So we're significantly reducing the level of virus in these susceptible tissues. So then we looked at vaccine durability. So this was done in C57 black 6 immunocompetent mice with a high dose of the vaccine in a prime boost strategy. And you can see here, following the boost there in the middle of this graph on the top, we see robust antiviral antibody titers, as we've shown you previously. And then if we extend this out to 18 weeks, we still see these robust antiviral antibody titers. And then we then challenged these mice, and we again see that these mice have low levels of viremia. These mice are not susceptible to any morbidity or mortality compared to the placebo group. So we're seeing robust, durable vaccine responses in this small animal model. So we published some of this data in 2017, and about very close to this as timing as our publication came out, another group, this is Drew Weissman's group from Penn, published a Zika virus prM-E vaccine with the mRNA-LNP platform. And so here's some of their data from the non-human primates. So what you can see is they also saw robust neutralizing antibody responses in the primates. And when they challenged these primates, they saw protection compared to the unvaccinated control group. So one of the hotly debated topics in the flavivirus field is what is the effect of pre-existing flavivirus immunity on Zika or dengue virus disease? And one of the prevailing hypotheses to why Zika caused such a strong pandemic when it hit the Western Hemisphere was that there was a large population of dengue immune individuals, and this was leading to an enhancement of infection. And in fact, previous data has shown that a heterotypic dengue infection, that is infection with a different serotype of dengue than what you're immune to, a heterotypic dengue infection can lead to enhanced disease in primates and humans through a phenomenon called antibody dependent enhancement, which I'll get into in a second. However, the data from the primates in the clinical epidemiology suggests that dengue immunity does not enhance Zika virus burden. And this is still a highly debated issue. And in some animal models, we do see that dengue immunity can enhance Zika virus infection. But overall, the primate data does not support this hypothesis. But what we do see is that Zika virus immunity can enhance dengue virus disease. So this is a paper looking at non-human primates where they demonstrated that prior exposure to Zika virus significantly enhanced peak dengue 2 viremia. And also we saw elevated cytokine levels that correlates with severe dengue. So we wanted to generate So just to give a little background on antibody-dependent enhancement and the mechanisms behind this. So if you look on the right of this graph, an ideal situation is you generate an immune response with a polyclonal antibody response which completely neutralizes the virus. You have your antibody completely coating the virus, and when the virus is taken up by these Fc gamma receptor bearing cells, the virus is completely neutralized. It's unable to escape the endosome and it's dead. However, on the left side of this graph, when you have incompletely neutralized virus, so in this case, you have antibodies that are not binding, have low antibody binding affinity, or you have antibodies that are binding to cryptic epitopes on the virus surface, these immune complexes are still taken up by these Fc gamma receptor expressing cells. The virus gets in the endosome, but now the virus is able to escape that acidified endosome, and the virus genome content is released into the cell, and you get continuation of the viral replication cycle. So increased viral entry and replication in the presence of poorly neutralizing antibodies. So what we set out to do was we set out to make a vaccine, a modified version of the vaccine, which we would not induce this antibody-dependent enhancement effect. So we know that antibodies targeting the highly conserved fusion loop epitope are driving antibody-dependent enhancement. And this is from years of work within the Flavivirus field. And here is shown the E protein dimer that sits on the surface of the viral particle. And I've highlighted here with this arrow is the fusion loop. And this fusion loop is highly conserved across the flavivirus, and it's non-neutralizing. So antibodies targeting this don't neutralize the virus. So we made a modified vaccine construct with the fusion loop epitope mutated. So the first thing we're looking at is an in vitro experiment in which we just transfect our mRNA into some tissue culture cells. And what you can see on the left is we're blotting with forming a Western blot with a monoclonal antibody that's just specific to the envelope protein. And what you can see is that we're seeing similar antigen expression level between the wild type and the fusion loop mutant version mRNA constructs. Now on the right, on this graph on the right, we're using a monoclonal antibody that is detecting the fusion loop epitope. So what we can see is that we no longer detect a band in the lysate from the mutant vaccine construct, so showing that the fusion loop epitope has been mutated. So first, we wanted to make sure that this vaccine has the same efficacy as our wild type construct. So here, we're just looking at serum neutralization titers with a high or low dose of the vaccine and a prime boost strategy. And what we can see is that both the wild type and the mutant construct are eliciting equivalent immune responses. And if we looked at the levels of the virus in the susceptible tissue, we see that both the wild type and mutant constructs are reducing the viral titers in both the central nervous system in the brain and also in the female reproductive tract. So the next thing we did is to look at antibody-dependent enhancement in an in vitro experiment. So in this experiment, we just incubate virus with the polyclonal immune sera, which here represented by the mixture of antibodies on the right. And this immune complex is then added to K562 cells. And these K562 cells are cells which express the Fc gamma receptor. So what you can see, and so here's some example data shown on the top right, with very low levels of the antibody, we see kind of a baseline level of infection. However, when that begins to increase, we see an enhancement of infection. This is due to the immune complexes being taken up by these cells and the virus replicating in these cells. However, at high levels of an antibody, we see neutralization of the virus as the antibodies are coating the entire viral surface, so we then are able to neutralize the virus. So when we look at our serum from our vaccinations, so in the red, we're looking at the the wild type vaccine. In the purple, we're looking at our fusion loop vaccine. This is serum from these vaccines, I'm sorry. You can see that we see this characteristic enhancement effect. So when the serum is very highly diluted, we see kind of a baseline level. But then at this mid-level, we see enhancement of the vaccine shown by this bell curve. And then at high levels of the virus, we're sufficiently coating the viral surface with our antibody and neutralizing the virus. We do not see this enhancement with the serum from our fusion loop vaccinated mice. So we are preventing the fusion loop mutation is abolishing the in vitro antibody dependent enhancement activity. And importantly, I think this data shows that the wild type vaccinated mice can lead to dengue enhancement. Now to test this in vivo, we're using a passive transfer model. So here we passively transfer our sera from vaccinated mice into a naive AG129 mouse. And this mouse is susceptible to dengue too. And what you can see, so here in red is mice that just received PBS. So they just got PBS, no immune sera. These mice do not display susceptibility to dengue too. When we give the mice in the black bars, these mice are given immune sera from a wild-type vaccinated mouse. These mice are now susceptible to dengue disease. In the open bars is mice that had received serum from the mutant version of the vaccine. These mice mimic the PBS, and they're not susceptible to the dengue enhancement. So this data shows that the Zika vaccine can enhance dengue pathogenesis in vivo, and the fusion loop modification abolishes the in vitro and the in vivo ADE. So I think this data is really interesting and shows some of the power of this mRNA vaccine technology and how we can easily manipulate specific antigens to shape the immune response towards desired directions. So the second thing we want to do now is look at congenital Zika virus. So as we said earlier, the greatest threat of Zika to human health is damage to the unborn fetus. And it's recently been shown that even asymptomatic infections can cause developmental defects in non-human primate models, which is scary to think about. So can our vaccine protect against damage to the developing fetus? So again, we're turning to our mouse model of Zika virus vertical transmission here. So in this model, so here on the left is, the top left is a wild type mice that gets Zika virus. And when this wild type mice, we know we don't see any Zika virus spread or any viremia. The virus is quickly inhibited. However, mice lacking an interferon receptor have high levels of virus. You get virus spread to the developing fetus and you get fetal damage. And this is shown by a lot of necrotic tissue in these fetus. We infect these mice at gestational day 6. And then this is looking at either 13.5 gestational day or 15.5 gestational day. And you can see that in the top right, we see high levels of Zika virus in the placenta and fetus when we knock out the interferon response. And we see placental insufficiency and a decrease in fetal size in these mice that have a deficient type 1 interferon response. So to test our vaccine in the pregnancy model, we are vaccinating female black 6, just wild type black 6 mice. Then 4 weeks later, we're boosting these mice. And 4 weeks after that, we're mating with a male wild type mouse. And then at gestational day 5, we're giving them 2 mg of our anti-interferon blocking antibody to limit that interferon pathway in the innate immune response. And then 1 day later, we challenge with the virus and then sacrifice the mothers at gestational day 13. And first, when we look at the maternal, at the mother, we see, as we expect, high levels of serum neutralization. So our vaccine is generating robust immune responses in the pregnant mothers. And this is protecting the mother from high levels of virus within the central nervous system in the brain. So we see high antiviral antibody titers and reduced viral load in the maternal organs. And importantly, we also observe that the vaccination is blocking Zika virus vertical transmission. So here we're looking at viral loads within the placenta and the fetus, and we see that we see low levels of virus in the fetuses in the placenta from mothers vaccinated with our vaccine. So we see lower viral burden in the placenta and the developing fetus. And furthermore, when we begin the images, so here's the placenta, we're looking at virus RNA via in situ hybridization. On the top is the placebo, and the bottom is from the vaccinated mothers. So in the placebo group, we see high levels of virus signified by this brown staining, and we zoom in, we see lots of virus in sort of the junctional zone within the placenta. the placenta from the vaccinated mothers, we never detect any virus RNA using these techniques. And furthermore, if we look at the total placental area, we see that the vaccinated mothers, placenta from the vaccinated mothers have a larger placental area. And we don't see the effects of the placental insufficiency that we see in the placebo group. So from this portion of the talk, we can conclude that, we can generate a prM-E encoded mRNA vaccine against the Zika virus. We vaccinate mice, we generate a polyclonal immune response. This polyclonal immune response can block Zika virus disease, we can block Zika virus vertical transmission, we can even induce sterilizing immunity, and some data I'm not going to go into today. But with this wild type vaccine, we can induce antibody dependent enhancement of dengue virus. With this mutant version of the vaccine, in which we've mutated the fusion loop, that conserved fusion loop epitope, we still see a blockage of Zika virus disease. We haven't looked at Zika virus vertical transmission, but we predict that since we were not seeing viremia with these mice, we're going to block Zika virus vertical transmission. But we no longer see the enhancement of dengue virus due to the antibody-dependent enhancement phenomenon. All right, so now I'm going to switch to some new data from my lab. And this is data that I started once I started my new lab shortly, just less than a year ago. And this is the development of a dengue virus vaccine using the same technologies. So as I've shown you before, dengue is very closely related to Zika virus. And dengue virus is actually made up of 4 different serotypes, dengue virus 1, 2, 3, and 4, which we collectively call dengue virus. So dengue virus is primarily spread by the Aedes species of mosquitoes, primarily the Aedes aegypti species of mosquitoes. And so here on the right on this map, this just shows the global distribution of Aedes aegypti. And you can see that this mosquito is spread broadly throughout the tropical and subtropical regions of the world. And that represents the spread or the threat of dengue virus. There's 400 million people infected annually with dengue virus. And 40% of the world's population are at risk to dengue virus infection. Now, an infection with dengue virus can lead to dengue fever, and this is an acute viral disease with varying severity up to hemorrhagic fever. Most individuals will not have any symptoms or very mild symptoms, but then some people can develop severe dengue, leading to death in some cases. Now, the highest risk of severe dengue is during a heterotypic secondary challenge. So what I mean by that is that individuals who are immune to one serotype of dengue are at higher risk to severe dengue following a challenge with a serotype of dengue other than what they're immune against. So a heterotypic secondary challenge. And this is due to the phenomenon of antibody-dependent enhancement. So there are several vaccines in the pipeline for dengue virus. And I'm highlighting the 3 vaccines which are the farthest along. These vaccines are either in phase 3 or have completed phase 3 clinical trials. And so I'm just going to go over these a little bit. The first is Dengvaxia. This is developed by Sanofi Pasteur. And this virus uses a yellow fever backbone. So all these viruses have the same concept. They're encoding the prM and the envelope protein of these viruses, or of the different serotypes of these viruses, in a live-attenuated backbone. So the Dengvaxia uses the yellow fever backbone, and you can see up here on the top, we've got dengue 1, 2, 3, and 4 all have the prM and E viral proteins in this yellow fever backbone. And this is a tetravalent vaccine, so it includes all 4 of these constructs, or these viruses in the vaccine. The second is a similar strategy by Takeda. This is TAC-003. But this virus this strategy is using the dengue 2 backbone. So you can see we still have the prM and E from all 4 different serotypes, but they all have the dengue 2 genetic background. And the idea here is that you're going to elicit cross-reactive T cell responses against the non-structural proteins that will protect against the dengue virus, where you don't have those same cross-reactive T cells from the yellow fever backbone. The third is a vaccine developed by the National Institute of Allergies and Infectious Disease at the NIH. This is TV003, and this uses the native background. This is a live-attenuated vaccine as well. This uses the native background. So you can see that dengue 1, dengue 3, and dengue 4 all have the native genetic sequence of those specific serotypes. Dengue 2, it has the prM and the E of dengue 2, but it's in the genetic background of dengue 4. But again, all of these are using the wild type, prM, and envelope coding protein regions. So Dengvaxia was the first vaccine that was completed phase 3 clinical trials. And this vaccine was licensed in 2015. And several countries began vaccination campaigns, and particularly the Philippines, where they had a really large vaccine campaign targeted towards children. And by November 2017, there was more than 800,000 individuals were vaccinated in the Philippines. It was later found out that this vaccination actually increased the risk of severe dengue in children. And then by April 2018, the WHO revised the guidelines. Now this vaccine is only recommended for individuals who are confirmed to be seropositive for a previous flavivirus exposure. And so right now, there's been a big blowback, lots of political ramifications of this vaccine and what has happened in the Philippines. And there's currently a massive outbreak of measles in the Philippines, largely because people have lost the trust in the government's ability to protect them, and particularly the vaccines that the government is proposing. So the vaccines that we develop against dengue and this flavivirus in general must elicit a balanced neutralizing response against all serotypes, and they cannot lead to any enhancement phenomenon. So we're proposing to use a novel strategy, the strategy that we just talked about before. This is the mRNA-LNP strategy, and really we want to limit the antibodies against the non-neutralizing cross-reactive epitopes, which lead to antibody-dependent enhancement. So here's the constructs that we're making. Again, we're using the same strategy where we're taking the membrane and the envelope protein coding regions. We're putting that behind a signal peptide. And this open reading frame is flanked by the 5' and 3' untranslated regions. This RNA is in vitro synthesized, and then we can enzymatically add a 5' cap and a 3' poly-A tail. And so we made both the wild-type version of this vaccine, or this construct, and a mutant version of the vaccine with the fusion loop epitope mutated. So we know that similar to the Zika virus, dengue virus, the strongest cross-reactive non-neutralizing epitope is the fusion loop, as shown here on this graph here, or on this image here of the virus. So this vaccine was in vitro synthesized and then encapsulated into a lipid nanoparticle. So first, to just show the construction and expression of our envelope protein. So here we are transfecting mRNA, either wild type or mutant version of the mRNA, into our tissue culture cells and looking at the envelope by Western blots. So on the left, we are looking at the domain. This is looking at a Western blot looking with an antibody specific to domain 3, so a domain within the envelope protein. And you can see similar expression levels between the wild type RNA and the mutant RNA expression of the envelope protein from these constructs. And in fact, we usually see slightly higher levels of the envelope protein in the mutant version of the mRNA constructs. So on the right, we're looking at a Western where we're using the monoclonal, which is specific to the fusion loop. And what you can see is that we're no longer detecting a band with the mutant RNA transfected samples. So here we're showing that the fusion loop has been mutated in this construct. Now importantly, we're still getting VLP secretion. So here we're taking the supernatant from these transfected cells, and we can concentrate that via ultracentrifugation, and we see similar expression levels, or similar VLP levels of VLP in the supernatant of these transfections. So the mutant has no effect on the VLP secretion. And when we look at the vaccine efficacy, so here we're vaccinating, this is just wild type black 6 mice. We vaccinated day 0 and collect the blood at that time. 4 weeks later, we're boosting, and then 4 weeks later, we're bleeding these mice to look at antibody neutralization, or virus neutralization. And so what you can see on the top right, this is just the pre-bleed, and you can see that both the wild type and the mutant at baseline, mice have no antibody protection. But then by day 56, on the bottom right, the combined data, both the wild type and the mutant vaccinated mice can neutralize the dengue 1. And dengue 1 is the construct that we use, so it's not surprising that we're seeing neutralization of the dengue 1. On the bottom left is the individual mice, showing the individual mice that were vaccinated. And unfortunately, that's all I can show you today. We're working tirelessly to look at the neutralization and the antibody-dependent enhancement activity against all 4 of the dengue serotypes, as well as Zika virus. And not only that, so all these experiments are underway right now. We're quantifying the T cell responses. So we know that T cells induced by these vaccines, T cells specific to the envelope protein, can be protective. We also have studies ongoing to look at protection efficacy. So in immunocompromised mice, we can vaccinate these mice, and we can look at the protection of these mice. And ultimately, we want to make a vaccine which is going to be protected against dengue 1, 2, 3, and 4. And so we are also generating constructs for dengue DV1, DV2, DV3, DV4. And we're going to compare these monovalent, bivalent, tetravalent vaccine strategies to see what generates the most robust response. And to conclude the talk, I think I've shown you that these nucleoside modified mRNA-LNP vaccines can elicit robust immune responses against these flaviviruses. These vaccines can be rapidly developed in response to emerging infectious disease and can be rapidly modified to enhance vaccine efficacy and safety. And I think this last point is how we can target the immune response towards specific neutralizing epitopes is really important, not only for enhancing flavivirus vaccine safety and efficacy, but also how we can make better vaccines against infectious agents which have been refractory to traditional vaccine approaches. This includes HIV, tuberculosis, malaria, and influenza. So this is a very exciting field. And so with that, this is my lab currently. It's really small. We just started less than a year ago. We're rapidly growing, so we've got a lot of work to do here, so I hope that we keep growing. The work that I did with the Zika virus was done while I was a postdoc in Michael Diamond's lab, and that was in collaboration with Moderna. And with that, I will take any questions.
Thanks, Justin. That was really interesting. We'll open it up for questions now.
So if you could just give me a little hands up, and I'll bring the microphone to you.
Thank you very much, Justin, for the presentation. I have just one question. So you mentioned at the end of your talk that mRNA viruses, this is a very good strategy to tackle those because they also have a very high mutation rate, right? You mentioned also briefly in one of your slides about the endocytic pathways. What's your take on targeting host function? So like endocytosis, transcription, translation, concurrently with...
So yes, that's something that's an interesting idea. I think that there's many ways that has not been explored how we can modulate the immune system as almost a co-vaccination strategy to enhance vaccine efficacy. That is certainly a huge avenue that our lab's trying to explore with various immunomodulatory mRNA or mRNAs encoded for immunomodulatory proteins. I think the strategy you're talking about would be, correct me if I'm wrong, would be to a therapeutic during an infection.
Yes.
It's a matter of catching the virus at the right time. Usually when people come into the clinics with these viruses, it's almost too late. By the time they present symptoms, most of the virus is cleared and we're seeing the secondary effects of the viral infection. So it'd be hard to get that as clear of a therapeutic approach, but that is something that would be interesting to do. It'd be fun to try.
Thank you. You mean also using small molecules and these mRNA vaccines?
It's something that we would love to do. Yeah, particularly if we could shape, for a viral infection, we generally want a Th1 immune response. So if we can drive that Th1 immune response to that specific and drive up a particular IgG2A antibodies, which are the best neutralizing antibodies against these viruses, that's something that we want to explore as well.
Okay, thank you.
Anybody else like to ask a question?
Am I allowed to ask one? My mic up? What do I need? I was just wondering, it was really interesting. What do you think? Do you think it's different target cells for mRNA vaccines versus other ones? Because this is something we struggle with in our lab. So I was really curious to find out what you were thinking.
So, so we've done some things with the GFP mRNA vaccine, and we see it within the lymph node and cells within the lymph node tissue. But we suspect there's some evidence with luciferase expressing mRNA vaccines that you can see it at the site of injection. So we think it's both. We think we're getting expression there at the site of injection, but we also expect that we're getting expression within the cells that are trafficking into the lymph node and inducing the immune response at that site. So. And that's important when you think about immune modulation, because when you have those immunomodulatory molecules, you want those to go to the site where you're eliciting this germinal center response and these kind of effects. So that's where it really comes important. When we look at VLP secretion, I think that what we really think is we just want lots of VLP. Once the VLP is secreted, it's going to be taken up by the local dendritic cells that are at the site of infection, but can also be taken up within the lymph nodes that are picking up things that traffic through the lymphatic system. So
Okay.
I don't know if it matters.
Interesting.
So we have a question here from Twitter.
Okay.
For you, Justin. So the question is, the serotypes for the flavivirus, are they constant and all defined, or can you expect to find new serotypes being generated by the virus like influenza viruses, where each year the vaccine needs to be optimized as the serotypes are different?
So we don't see kind of like serotype switching. These 4 dengue serotypes are relatively constant. I mean, we do see mutation rates, but the serotypes are constant. The protection against one serotype is expected to be protective against that same serotype down the road.
So we can take some questions from Twitter now. So we have a few for you, Justin. So this is from Brazon Therapeutics. It says, great talk so far, and this is in relation to the targeting question. Do you have any thoughts on targeting microglia for central nervous system infections?
Yeah, so we haven't looked at specifically targeting specific immune cells, and I think that's a great point, particularly when we think about delivering antivirals for some of these neurotropic viruses, which includes West Nile virus and Zika virus to some extent. So I think that is a fantastic avenue that we'd like to get into, but we need to talk to the formulation experts to figure out how to do that.
So we have another question here for you, Justin. As we saw that you've used an ionizable lipid, how much is the encapsulation efficiency and the practical loading of the mRNA per weight of the nanoparticles? Also, are these lipids used similar to the MC3 class?
A couple things on that. I want to make sure I answer your question, but I want to get a couple other points in there too. So our strategy is different than what we just heard about today. We're actually trying to limit that innate immune response because we want to drive the gene expression from our mRNA. And it's known that innate immunity will limit the transgene translation of our mRNA, endogenous or exogenous mRNA. Now, to answer your question, sorry, let me go back. So we're using a little bit different parameter. We're using a lipid nanoparticle. So we're using a cationic lipid, which is coating our nucleotide RNA, and then we're coming in with the other lipid mixture that coats the entire the cationic lipid mRNA complex. So we are actually getting really high encapsulation efficiency. We're getting around 90% encapsulation efficiency, and we're not losing we're losing very little of our mRNA that we're putting into the system. We're using the same benchtop nanoassembler that Yvonne just talked about. So our encapsulation efficiency has been really good, and so we're not losing much. In terms of the weight of the mRNA to the lipid nanoparticle, it really varies. So our mRNA, we're using several different mRNAs. The ones I talked about today are roughly 5 to 6 kb. We put in 9 kb. We put in things down as low as 800 base pairs. So we see similar sort of encapsulation efficiencies regardless of the mRNA that we're using, and so we don't think that matters too much.
So we have one final question for you here, Justin. Has your lab ever immunized the same mouse with multiple different dengue viruses? If so, would you see any problems with this strategy?
That's something that we're currently doing right now, and I think that's something we're exploring is what is the best way to generate protective tetravalent responses? Is it putting all the vaccines at once, which is the current vaccination strategy that's in the clinics, or is it kind of a reiterative approach where 1 versus 2? And I think that's a really interesting area of exploration that's going to take some work.